First, I would like to thank the organizers for giving me the opportunity to make this talk. I'll present here a summary of what has been done on quantitative resistance to blackleg in oilseed rape. As it has been presented, there are two types of resistance to control blackleg. These resistances operate at different stages of the fungus life cycle. Qualitative resistance controlled by measured genes is effective from young plant stage onwards. It prevents leaf lesion development if resistance genes correspond to avirulence genes present in the fungal population. Quantitative resistance is controlled by many QTL for quantitative trait loci, each having a low to moderate effect on resistance. This quantitative resistance limits the necrosis development at the crown of the plants. Quantitative resistance was shown to increase the potential for durability of measured genes, as exemplified here with RLM6. An experimental design set up by Hortense Brun actually mimics what happened in real cropping conditions. 4 series of recurrent selections were with crop residues of Urol RLM6, Darmor RLM6 were performed. Urol is a susceptible variety, Darmor is a variety with quantitative resistance. RLM6 was introduced into oilseed rape from Brassica uncea. Each year, crop residues were harvested on each genotype and used as inoculum for the following season. Number of leaf lesions and crown canker were assessed. Here you have the disease index evolution along years, with Urol RLM6 residues. Virulent population increased rapidly, and there was rapidly no significant difference between Urol and Urol RLM6. With Darmor RLM6 residues, virulent population increased more slowly, as shown here. Urol RLM6 remained less susceptible than Urol for many years. It's only after 8 years of recurrent selection that virulent isolates increased. This could be explained by the fact that even if virulent isolates appeared, their multiplication was reduced thanks to the quantitative resistance. And even after the population evolution, quantitative resistance controls the disease. As we see here, a low disease index on Darmor and Darmor RLM6 was observed. In addition, after recurrent selection with Darmor residues, no adaptation to quantitative resistance was shown. Then, we recommend to use quantitative resistance alone or in combination with RLM genes in order to control blackleg disease. In parallel with these studies, we initiated genetic analysis of quantitative resistance. We began with the study of 2DH population derived from Darmor. Darmor is a double low line derived from Jet9, which was a well-known variety in Europe with a high level of quantitative resistance. Each population was evaluated during 2 years. A number of QTL were identified according to year and genetic background. Some of them were common to the two populations. The YDH population was evaluated in different environments in order to study the stability of the QTLs. It was first evaluated in France and UK over 5 years, which showed 6 stable QTLs on A2, A7, A8, C2, C4, and C8. 3 QTLs on A2, A4, and A6 showed interaction with environment. Then the YDH population was evaluated in Australia, which showed that 8 QTL on A2, A6, A7, C1, C8, and C9 were detected in various environments. So this showed that some QTLs could be stable over countries and in consequence stable over Leptosphaeria maculans population, which could be different between the countries. We then wanted to know whether Darmor QTL were present in other resistant sources and if we could find new QTL. We used connected design with 4 populations sharing the same susceptible parent, Bristol. We chose resistant parent without effective RLM genes in order to study only quantitative resistance. populations were evaluated for 3 years, which showed a higher effect. You can see here the disease severity was much lower in 2009 than in 2010, for example. QTL detection was performed in the 3 populations. From 1 to 8 QTL was identified per year, and these QTL explain from 3 to 31% of the phenotypic variation. Here you have the schematic representation of the linkage group, and each rectangle represents 1 QTL. When we compare the location of these QTL, we found that 8 QTLs were detected over 2 or 3 years. Then we compared the QTL between the different populations and showed that 10 QTL were common to the different resistant parents. Some of them were common to Delourme QTL, but other QTL were derived from the other resistant sources and were different from the one of Delourme. We can see here also on C1, for example, that some QTL were specific to one resistant source. When we looked at the genomic localization of the QTL and took into account the collinearity between the chromosomes, we found that some QTL might be located in homologous regions, as we see here for these 7 pairs of QTL regions, located on A1C1, A2C2, A3C3, and so on. In order to have a more complete view of QTL regions and alleles in winter oilseed rape, we initiated genome-wide analysis studies on different panels of winter oilseed rape. We enriched genotyping data on biparental population and panels using the 60K SNP array. QTL detection was performed per year, or on blob estimates that gave a phenotypic estimate over years. Here you have the schematic representation of the results with all the chromosomes indicated on the top of the figure. Each line are the QTL detected per year or on blob in dark colors. The QTLs detected in the biparental population are indicated in orange or red, and the QTL detected through genome-wide analysis are indicated in blue. 16 QTL regions were common to at least 2 biparental populations. 8 regions were significantly associated with quantitative resistance in the panels. 5 regions on A6, A8, A9, C1, and C4 were common to linkage mapping and association mapping. When we looked at the collinearity of the chromosome, 8 pairs of homologous QTLs were identified. Due to the complexity of this trait, we investigated the possibility to make genomic prediction of resistance level with a set of markers. Kumar showed that 3,134 SNPs associated with resistance provided high prediction accuracy of resistance. Of course, this prediction equation should be adapted to panels and genetic background. From year to years, more and more studies have been published. Many genomic regions were shown to be involved in quantitative resistance. Some QTL were derived from Delourme, here in green, and other QTLs were identified in other resistant parents, here in orange. You can have a look to Amas et al. 2021 for a review from which this figure is taken. In all these regions, many potential candidate genes and functions might be identified. They might correspond to resistance genes analogs, genes involved in defense pathway, or other unpredicted functions. In parallel, some studies were initiated to better understand how QTL operates and which mechanism might be involved in resistance. Yongzhu Wang et al. In different studies showed that quantitative resistance mainly operates in the stem of the plants. She performed different inoculation methods. First, after leaf inoculation, she studied the growth of Leptosphaeria maculans along peciole. Second, after peciole inoculation, growth of Leptosphaeria maculans in the stem was assessed. In a first experiment, 2 varieties were compared. Delourme with quantitative resistance and Ural as susceptible variety. After leaf inoculation, no difference was found for fungal growth in the peciole, but stem tissue were less extensively colonized in Darmore than in Ural. In addition, Yongju showed that at 25 degrees, the expression of resistance was reduced, which should be taken into account in the context of climate change. Then, 2DH derived from DYDH population were compared. A30 is very susceptible and C119 highly resistant. This showed again highly significant difference for growth of Leptosphaeria maculans in the stem, and showed also significant difference in area of leaf lesions and distance grown in the peciole after leaf inoculation. We then performed a QTL analysis in DYDH population with leaf inoculation. We studied the growth of Leptosphaeria maculans in the leaves and in the pecioles. Many QTL were detected, of which only one on A02 co-localized with a QTL detected in the field. This confirmed that most part of quantitative resistance operates in the stem, and only a small part could occur in other organs, and this depends on genetic background. We developed a new inoculation method to study quantitative resistance action in the stem. At 3-leaf stage, we cut the peciole of the second leaf and put 10 microliter of conidia solution on the peciole section. Leptosphaeria maculans migrates to the stem and necrosis appear 15 to 20 days post-inoculation. Here you can see external and internal symptoms with typical necrosis and tissue blackening. We coupled this inoculation method with quantification of Leptosphaeria maculans DNA through qPCR to follow the growth of the fungus. We confirmed the significant difference between Darmore and the susceptible varieties Ural and Udall, as shown here for the length of external symptoms. Interestingly, this showed that the speed of symptom development was different according to genotypes. It was more rapid on Ural than on Udall, despite Udall is more susceptible in the field. But if scoring is performed at later stages, for example at 50 or 60 days post-inoculation, we can see higher symptoms on Udall compared to Ural, as seen in the field. We sampled stem section at the inoculated peciole insertion site and just below, and then quantified fungal and plant DNA through qPCR. This confirmed the resistance of Darmore with low fungal amounts, and confirmed the different speed of fungal growth in the stem of Ural and Udall. At 42 days post-inoculation, the amount of Leptosphaeria maculans was much higher in Udall, which provided a good proxy of its field behavior. We applied this method to near-isogenic lines that were derived from DY population. Through back crosses, near-isogenic lines with a single Darmore QTL in Udall genetic background were obtained. We had previously evaluated these near-isogenic lines in the field in different locations. This showed a significant effect of A02 and A09 QTL. When we used our cat peciole inoculation method, no difference was observed on symptom development, but a significant difference was found for Leptosphaeria maculans DNA amount. This confirmed that our method can be used for quantitative resistance study and even for low QTL effect assessment. In order to go towards the understanding of oilseed rape Leptosphaeria interactions and towards the understanding of the mechanisms underlying quantitative resistance, we ran a metatranscriptomic project with Biogère unit with Mylène Balédan and Thierry Rouxel. This consisting in studying oilseed rape Leptosphaeria interaction at different stages, using Darmore, Dwarf Darmore as resistant varieties and Bristol as susceptible variety. We performed cotyledon sampling PCL sampling after leaf inoculation, stem sampling after PCL inoculation, and stem sampling in the field at different times of the season. In addition, in vitro and residue samplings were performed to complete the life cycle. This wide study allowed us to decipher the life cycle of Leptosphaeria maculans and Biglobosa. I will not detail the results here, but Elise Gay identified different effector waves that correspond to specific infection lifestyles or to tissue-specific genes. Our objective was also to decipher mechanisms underlying quantitative resistance of Darmor. Our questions were: how do Darmor and Bristol respond to Leptosphaeria infection? What is specific to Darmor response? And are there some constitutive differences between Darmor and Bristol? And what is their relationship with QTL? For this, an experiment was run in control conditions with the 3 genotypes and 2 Leptosphaeria maculant isolates, GN2 being more aggressive than NDT4. We used the cat PCL inoculation method, scored the symptoms, and sampled the stems at 2 time points, 14 and 28 days post-inoculation. Here you have the length of external necrosis at 28 days post-inoculation. We clearly see the difference between the susceptible and resistant varieties. We also see the higher aggressiveness of GN2, especially on Bristol. We also sampled stem section in the fields on 2 genotypes, Dwarf Darmor and Bristol. Here you see the disease index which discriminates the 2 genotypes from the end of April. Stem samples were collected at 7 time points along the season. First, we studied the response to Leptosphaeria maculans infection, and we looked at the genes regulated in Bristol, Darmor, and Dwarf Darmor in common to the 2 isolates. 56,980 brassica-napus genes were expressed in the stem. Here you have the number of regulated genes after infection in Bristol, Darmor, and Dwarf Darmor at 14 and 28 days post-inoculation. This showed an important core response common to Bristol and Darmor. Many genes were specifically regulated in Bristol, and only few genes were specifically regulated in Darmor. In order to characterize this response, we performed an inventory of genes involved in defense, resistance, specialized metabolism, and so on. For example, we saw a clear salicylate response. Most of the genes involved in salicylate pathway were induced after Leptosphaeria maculans inoculation, as shown here at 14 days post-inoculation. And this induction was even higher with the most aggressive isolates, GN2. We found that most genes involved in aliphatic glucosinolate pathway were downregulated, and most genes involved in indolic glucosinolate pathway were induced from 14 days post-inoculation. Again, we saw a higher induction of indole glucosinolate genes with the most aggressive isolate, GN2. These results were confirmed with quantification of indole glucosinolates in the stem by liquid chromatography. This showed the accumulation of indole glucosinolates after Leptosphaeria maculans infection in both genotypes, and even a higher accumulation was found in Darmor. We also found an induction of genes from indolic phytoalexins biosynthesis. We investigated the genes involved in phenylpropanoid pathway and found that lignification increased along developmental stages, as shown here for C4H genes. There are 10 copies of this gene in brassica-napus genome, and we see here their expression level in the field from November to May in the upper part of the figure, and in control condition at 14 and 28 days post-inoculation in the lower part of the figure. We can see the expression increase of most of the genes along developmental stages. No clear difference was observed between Delourme and Bristol in control conditions. A higher induction is even seen in the field in Bristol. Then, there is no clear implication of these genes in quantitative resistance. We then compared the response of the resistant genotypes to the one of Bristol and looked for genes upregulated in Delourme and dwarf Delourme compared to Bristol. We have here the relative expression of some genes showing differences between susceptible and resistant genotypes in control condition on the left and in the field on the right. For each condition, we have Bristol on the left and Delourme or dwarf Delourme on the right. These genes correspond to genes coding defense protein, genes involved in signaling pathways or specialized metabolism, or genes with unknown functions. We then investigated the constitutive difference between the genotypes. It means differences observed in mock-inoculated plants with water in absence of Leptosphaeria maculans. Here we have the number of differentially expressed genes between Delourme and Bristol, or between dwarf Delourme and Bristol at 14 and 28 days post-inoculation. This showed that many genes were constitutively differently expressed, and especially a high number of genes were differentially expressed between dwarf Delourme and Bristol. All these genes were localized in relation to the position of the QTLs, which were previously identified, and this led to many potential candidate genes. They are further investigated, but it's really a tough work due to the number of genes and QTL regions. In conclusion, quantitative resistance is a highly polygenic resistance. Many QTL were identified, which can be dependent on environments and genetic backgrounds. But some stable QTL were found, which is very interesting to get stable resistance across environments. Some were obtained on how quantitative resistance operates and in oilseed rape response to Leptosphaeria maculans infection in compatible interaction, either in partially resistant or susceptible genetic background. Many potential candidate genes were identified within or outside QTL regions, which either show differential response to infection or show differences without Leptosphaeria maculans infections. There need further studies to identify the most relevant genes using different methodologies, such as studying the differential expression in near isogenic lines, studying their sequence polymorphism, or obtaining mutants. In parallel, we perform metabolomic studies to identify potential metabolites involved in response to infection or in quantitative resistance. This is done by quantification of targeted metabolites through LC-MS/MS, or by the study of spatial distribution of metabolites on stem sections through MALDI-MSI. MALDI-MSI-based metabolomics allows highlighting metabolic responses that are specific to necrosis surrounding tissues in infected stems. This needs cryosection of the stem, laser excitation of the section after a specific matrix has been applied, and mass spectrometry imaging on all the surface, which allowed spatial segmentation and spatial distribution study of molecules. These molecules are identified through their precise mass over charge ratio. For example, we can see here spirobracicin, which is accumulated in the vicinity of necrosis area, or methylmethoxyindole 3 carboxylate, which is detected in undamaged stem vessel structures. We have shown that quantitative resistance can be used alone or in combination with RLM genes in order to ensure potential durability of the resistance. In France, in relation with the variety registration rules, the number of resistant varieties has much increased with time, and there are mainly highly resistant varieties now. From registration and post-registration analysis, we know that both types of resistance are used. Varieties carry either only quantitative resistance or RLM genes and hopefully quantitative resistance background. As we have seen, quantitative resistance is a very complex trait for which genomic selection would be interesting to use, with, of course, establishments of the prediction equation in genetic backgrounds that are used in breeding programs. This needs multi-environment evaluation to get relevant results, and this would need, I think, further phenotyping improvement to get easier or more precise resistance assessment. This can be done with different methods such as qPCR or image analysis. I would like to thank all the colleagues who participated to these studies along all these years, in my research unit at IGEP, in Biogère unit in Grignan, in the Genoscope at Évry, and on Protein platform in Rennes. I also thank our experimental unit and metabolomic platform P2M2, and of course the funding. And I thank you very much for your attention.